Hi everybody, and thank you for joining our online panel discussion today. So the focus of today's panel discussion, the topic is going to be centered around data visibility and ownership when working with external partners. So kind of the conversation will kind of touch on three core topics. The kind of current challenges that we faced within the industry with data visibility and data ownership. Then a deeper dive into some data ownership perspectives and discussions around kind of the utilization and compatibility for raw versus processed data. And then we'll wrap up with more of a discussion around kind of storytelling with data, the importance of contextualization, not just information management, but data knowledge management too. So just to kick things off, we shall introduce our panelists. So today I'm delighted to be joined by our digital products lead, Bret Huff, and two of our senior analytical scientists here at APC, Sabine Harrison and Pól MacFhionnghaile. So perhaps Paul, you might start us off by telling us a little bit about yourself, your background, and what kind of your current role in APC, what does that entail?
Yeah, so I'm Pól MacFhionnghaile. I am a senior scientist here at APC, in the analytical function, mostly with solid state analytical. I spend a lot of time looking at graphs, looking at data, and trying to contextualize different techniques to tell a full story of the material that we're looking at.
Okay, great. And Sabine?
Sorry, Sabine, I believe you're muted.
I'm not here. Yeah, I'm not. Perhaps Bret, you could jump in there while we're resolving the technical difficulties there with Sabine's mic.
Yeah, good morning, good afternoon, everyone. I'm Bret Huff. I'm the digital products lead at APC. Before that, I was at Eli Lilly company for almost 30 years and was both in development and discovery there, small molecule discovery and development. And I've had a real passion throughout the years of, trying to help scientists digitalize their data, organize their data, and try and be able to tell their story, their development story efficiently. As part of APC, I've been working with Aaron mostly to design and build iAchieve. It's a collaborative, transparent CMC collaboration platform that we use with our clients.
Great. I think we should have Sabine now.
Hello, I'm Sabine. I'm working with Paul together in the analytical function, and for me, data sharing analytical. So I'm really excited to talk to you a little bit more about data transparency and data ownership today.
Okay, brilliant. So then maybe what we'll do then is get kicked off with the poll results that we had from our poll. So we ran a poll on LinkedIn and the lead up to this event to be able to get a steer kind of on what the industry felt were the biggest challenges facing the pharma industry when it came to achieving data visibility across the CMC journey. So we can see kind of from some of the responses, we kind of got a tie between responses for integration between CMC stages and insufficient data governance. The minority chose lack of data standard formats as being the biggest challenge. So the siloed systems and fragmented data were by far the most popular response that we received. So maybe we can start by kind of going into that a little bit more and getting our panelists, perspectives on, based on your own experience, perhaps, what are the biggest or most common data visibility challenges you faced throughout the CMC development journey? So maybe we'll start with Bret.
Yeah, so first of all, I totally agree with this ability to be able to find and organize your data as being a number one problem. I think ironically, there are almost too many ways to communicate. If you think about all of the ways that we communicate and all of the places that we store data, SharePoint, team sites, Box, we communicate through email, we communicate through instant messenger. There's almost too many ways for data leakage and decisions that occur outside of the team, that becomes a real problem in our ability to be able to reconstruct that development history and that development story over time. And so, one of the problems that really I think is a tough one for scientists is that they have to spend a lot of time curating that data and finding that data, taking care of that data that's really, in disparate locations, all across the enterprise, and especially in working with CDMOs or CROs, a lot of that data and that information can be even outside of the organization. So yeah, I agree, it's a real problem.
Great. And Pól, would you think that the data being siloed and fragmented is probably based off your own experiences as well as an analyst in a number of different areas? Would you think that would also be one of the largest challenges, or have you encountered a more common challenge to data visibility?
Okay, there we go. I think I've got signed back. Aaron, you hear me?
Gotcha.
Yeah, absolutely. There has traditionally always been a problem of data being siloed and fragmented. Traditionally in analytical science, chemistry, most disciplines, I mean, as we go through PhDs, as we go through our own research, we generate our own data. We keep our own data, we look at it, we create our body of work, and then we throw it out. But when we go into a more team-based system like the pharmaceutical industry, then we do need to be able to connect with each other. We currently have a system in our labs where I can get a full visual of what's occurring with everyone. I mean, when you contrast that to where I was 15 years ago of not knowing what my benchmate was doing or what they were analyzing or what was happening, it's absolutely amazing now. It's fantastic. And I'm pretty excited to see where things are going to go.
Brilliant. And then Sabine, what would your perspectives be based on your experience? Would you agree with Bret and Pól in terms of that being a challenge, or?
Yeah, it's definitely a challenge that everybody, like the smaller the lab is, the more computer-based you are, rather than having a server where you have everything, at least from, if you have different analytical instrumentation where you can put it all into, at least for one instrument type, or maybe some instrument types on one server. So it's all that already makes it easier to find. And in that respect, that would probably go a little step further still, where I say, like, especially if the bigger the organization is, the more structure you have even to name samples and to have a uniform sampling way, a uniform way of naming your samples. Because as Pól was reminiscing about his PhD, he probably just called it a brown slurry A and brown slurry B, whereas this makes it really difficult to also have this whole data visibility, because maybe in a year's time it doesn't know what that brown slurry actually was. So I think that again makes it really important.
I'm quite attached to all my brown slurries. I keep them in a shoebox back home. I mean, yeah, I never leave without them. But in saying that, I mean, like, I think you really hit a massive point there. I mean, I was just one person doing a study, whereas if you go into an organization that is decades old, the cultural change that must occur in that. I suppose, Bret, you might have some experience in that. The fact that it isn't just a lab going, this is how we're going to do it, but there has to be an organizational step in. And the burden has to be carried out through everyone, not just people in the labs. Would you be able to speak on that a bit?
Yeah, it's a great point, Paul. And you know, when you're working in a big team environment like we do in the pharma industry, it's not only cultural change for a person, you know, who comes from that PhD lab where they're used to not really having to collaborate. It's their data, it's their science, or and you know, moving into industry where it's a team's data, it's a team science, it's a team problem. But then you've also extended that now to even different organizations, either within the company or external to the company. And they all have their own data cultures and their own way of thinking about whose data it is and their own processes for approving data or moving data into different kinds of storage or in how they interpret data. And so it's a huge cultural challenge as well to get everybody on the same page. Of understanding where the data is, how the data is treated, and how it's shared, you know, and I think that's one of the things that, you know, we've really tried to do at APC, which is very exciting, is to think of the data as really being not only the clients but the teams, and to have it be very transparent and to have it be shared across the team. It's really, I think it's really exciting.
And I think that there's something so important about the fact that, in a weird way, the data not being mine makes me care about it more. You know, when I'm working with a client. Back in my PhD days, I was very cavalier. I remember at one point sifting through bins to get a hard drive because I didn't save something correctly, and having to just root it out and, you know, retrieve data. And again, I did that because it was my data. I didn't care. Like, you know, it's only going to be in a graph in a PhD. It turns out being vital, and terrible, terrible data management from a 20-year-old Paul, unsurprisingly. But yeah, when it becomes the client, it becomes this beautiful product that you produce, that you curate, and where you can show your client and say, this is this diffractogram. You know, here is the pattern that I produced, and it's a nice PDF, but this is the raw data. Here's the context, the experiment, this is the raw data.
Yeah.
You get to handle it, you get to do with it what you want. It's not limited by my observations and by my assumptions on what it means. You know, I will have my assumptions and observations in the experimental section, but the client gets to travel a bit further. And I think, Sabine, like, you know, when it comes to the LC side of things, that can be a little more vital. I mean, I've seen reports where you're looking at this tiny PDF graph and trying to see if there's two peaks one. I mean, would you be able to go a bit more on that?
Exactly, and it's, I think what Bret was also saying, it's the culture really, because obviously it's not just the culture of how the naming conventions that I mentioned, but it's really the culture that allows people to discuss them and share them. And yes, especially like for me, for chromatography, it's really important that you don't just share a PDF and that's one person's interpretation. And I even like to think back to when I was in my PhD and people were still printing on paper with a felt tip pen that was moving over, where you literally had one go at integrating something because you had to actually cut it out of the paper and then weigh that because you didn't have peak areas and automatic integration. We've come such a long way, and people may think that that's not happening for 25 years. Like if you've been in a university lab recently, that's still happening, really. So it's just we've come such a long way, and that's really the lovely culture as well in collaborating and sharing the data and being really transparent about it, because it just gets so much more out of it.
Yeah, I mean, when you consider, I mean, yeah, maybe back in those days, you might get a credit or a module for arts and crafts, and you're losing out on that. But being able to have your data and look back at it, yeah, I'd sacrifice that little cert saying I did arts and crafts and got my scissors out and everything. Like, you know, that just, it baffles me. It's amazing. It's revolutionary. Like,
and,
you know,
and to expand a little bit further onto that kind of topic around data ownership and perspectives, maybe I might put a question. So Sabine, you kind of touched on it a little bit earlier on, to the whole idea of like, what, in your opinion, what do you think is the real differentiator between kind of just facilitating data ownership, because after all, the data that we generate for our clients belongs to our clients, and really enabling effective data ownership? Could you maybe comment on that?
But as we discussed a little bit already, it's like there's ways of sharing data, and for me, data is really the raw data where I can go back and look at it and see how it happened, how the result was generated. Data for me is not a PDF that Paul has already mentioned. It's really the raw data where I can go back, I can zoom in, I can overlay various samples with an ironic and overlay with a standard, or I can even sift back to do some trending and overlay and see how peak shapes have moved, because that's really something really important. The peak shapes have changed. So for me, that is really one critical thing, where how data sharing is really the raw data. It's not a PDF. A PDF is an interpretation of the one person who generated the PDF, and that's not effective data sharing. That effective data sharing is the raw data.
Yeah, I think just picking up on
I couldn't agree more.
Which I totally agree with. I think even I like to think about that data as being alive still alive through history. You know, once you've interpreted that data and made a PDF of it and shared it, it's kind of dead. You know, you can't go anywhere from there anymore. You know, it's not open to reinterpretation. It's not open to new information that can be applied to it. And so I think keeping that data alive, by being able to access that raw data in its structured form is really valuable throughout history, you know, and throughout whenever you might need that data again.
Exactly, because your question may change, and you may suddenly be made aware that that teensy-weensy little peak at the end of your chromatogram that you dismissed because it was teensy-weensy suddenly becomes really critical, because it might actually be really, really toxic. So it's really important to be able to go back and have a look if that has always been there, and was it always the same size. So that makes it really crucial. Sorry, Paul.
Oh yeah, no, I was like, I think everyone here would agree with that exactly. And then there's the other aspect of trying to look forward. As you mentioned, you know, you might look at that teeny weeny peak that might be crucial, but there's also a bit more. There's also the advancements. So if you have all data that can be read and accessed and structured correctly, then you can lean on that and actually start seeing variations that are occurring and linking it back to process changes. So you end up with this historical bank of data that you can mutate into quality by design approaches, really. You know, in where, say, particle size, there's a slight deviation, it's still well within the spec, we're seeing drifts occur, and you'll be able to relate it back to what actually caused it. So then when something else is drifting in another way, you can go back and fix it. You can actually do robustness tests, as Bret said, when the data is still alive. You know, when that data is still alive, you can always really take from it and let it grow. And whatever models you're building of your plant, of your processes, if the data is still alive, then you can always find more and more information. But when you close it, it's gone.
Yeah.
And I think that linkage relationship that you mentioned there, Paul, as well, is really critical. It kind of nicely leads us to the final topic that we're going to be looking at in terms of storytelling with your data, because naturally, as we've already mentioned, the context around a single experiment is critical, but sometimes it's the relationship and the linkages between a set of experiments and being able to compare the context and the conditions between them that really reveal those insights. Because I mean, today's data, you may not know its full potential until you've run tomorrow's experiment. So it's really about making sure that you have the data captured in like a future ready state, so that if you do need to bring it back up and to drill back down into it and kind of, as you said, Sabine, overlay it with other things that you've seen, you don't run into any barriers there. You have it all in a usable format that allows you to continue writing that story, as opposed to just, you know, a freeze frame, as we'd said, the PDF kind of gives you.
Exactly, and it's just, especially with all the advancements in statistics, even like there is literally packages out there where you can put your raw data in, and it's literally trending you a single peak if you wanted to. So that's stuff that you wouldn't have been even able to probably 5 years ago. So who knows where it's going to go in the next 5 years? And it just takes some of that human element and that human interpretation out as well, which makes it even more critical to use our raw data to the best, and as both Paul and Bret said, not freeze it in time in a PDF, because that you can't do anything with it. Well, you can frame it and hang it on the wall if you want to, but that's probably all it might be good for.
It can go with your arts and crafts cert.
Yeah.
No, but I think that you're quite right, and like we can never really tell what the next 15 years are going to bring in innovation, particularly when it comes to technology and data interpretation. I mean, right now we're doing a webinar, a video call with so many people. This would have been impossible when I was 10 years old. When you look at statistics and doing data interpretation, when I was doing my PhD, it was revolutionary that I was being able to process thousands of spectra. Now we're in hundreds of thousands and millions. I mean, so, on a desktop, this is not using a supercomputer or renting out like your server time from Amazon. This is using our desktops and laptops. But the key in keeping data alive is being able, keeping the legacy there, is having the raw data being accessible, that we don't manipulate it, that we don't close it off, that we don't lock it behind barriers and, I don't know, make a PDF while the raw data rots away in a hard disk somewhere in somebody's basement. The raw data, when I'm dealing with a client, is part of a product, and it is something that the client can use, and quite often, hopefully they will use it, but we give them that ability to use it, and that is key.
Yeah, I think that's actually touches on a really important point in terms of the raw data versus processed data. Naturally, to derive and derive your interpretations, you need to process the data, but making sure the raw data is available as well. It kind of gives the full story. You're not just getting a synopsis, which is kind of, or you're not just getting the one person's take on it. Yeah. So for storytelling with data, I mean, looking at that as a topic, what would your experiences be kind of internally here? How we use and how we share data internally and how we tell a story? Maybe put that to Bret.
Yeah, I Aaron, you know this well. I love this word around storytelling, this idea of storytelling. I think scientists are great storytellers, and I'd love to hear their stories. Why did they go down this pathway? How were they thinking about a problem? How did they solve that problem? And you mentioned this word contextualization earlier, and I think having that data in context and being able to tell that story then, to know why they collected the data, where is the data, how can I reuse that data for the future, and how can I ultimately tell that story of how I solved a problem. What is the development history of this? What's the story that I want to tell a regulator or I want to tell a manufacturing site? I think having that data, like we've talked about, alive and able to find it and have it in context, so I know why this data was collected, not just where it is, but why was it collected, I think is one of the things that internally at APC we really do a good job at doing, to contextualize the data and to be able to tell that story to our clients and ultimately help the clients tell that story to regulators and manufacturing sites.
And it's really important as well, because sometimes people just think about a specific instrument or a specific result they're looking at, but the orthogonality, by having loads of different measurement data together, that's really what helps you frame that story and tell the story, that you don't just have a single result on a single computer or on a single hard drive even, but that you have all the different data of all the different measurements that you have taken together, because it's the sum is more than the parts of it in that case, really, because each one gives you a snapshot of your sample in time, but to actually take all those measurements together really helps you tell a story. And I know Paul loves those orthogonal methods and how you can tell a lovely story with that.
Yeah, absolutely, Sabine. Like, and in the guise of storytelling, when you're looking at different methods, particularly like when you're looking, we'll say at solid state or thermal analysis, particle properties, different techniques are like a paragraph of that story. And you need the entire complete set of paragraphs to finally be able to tell that full story. And that's where the great scientists here at APC come in, that I get to hang around with, which is always fantastic. I think that you said it quite well there, Bret. We are good storytellers, and we'll always tell that one paragraph very well. And then we get to the next paragraph, and we get to tell that story, get to the next, and we get to tell that story until we get the full story and beginning, middle, and end. We have it all, and then we get to deliver it to the client. And of course, they might turn around and say, we don't agree with you. And we get to say, well, this is where we came from these conclusions. We didn't pluck them from the air. These are where they came from. And they may take the raw data, look at their historical evidence, and come back with something else to add to that story. Doesn't kill it, it adds to it. It's always a collaboration. And then we can carry it further. I love the idea of it being a story. Otherwise, it becomes an argument when conflicting ideas hit. And it's just a lot easier.
Yeah, exactly. And it's sometimes that can be useful. I mean, to be able to make sure that your data is robust and that your conclusions are sound. It's important to be able to give people the ability to challenge and interrogate that data. But I think we might just want to probably come to wrap up and come to closing remarks. I mean, I know that we could very much talk about this topic for another hour or longer, but just to be able to maybe wrap things up, and then also kind of consider to the audience as well, if they're interested in learning more about kind of the information, getting more information in terms of how we're tackling the issues of kind of data visibility and effective data ownership through radical transparency. We like the platform that we've developed and have grown within the Medicine Accelerator to do this, and it's a tool that we didn't touch on in this panel discussion, but maybe in future we will do. Is iAchieve, and it's something that we are finding have transformational effects to really help tell that story and make the data future ready. So there should be an email on your screen there if anyone's interested in finding out more about how we're tackling this data visibility challenge and kind of really in driving data ownership to our clients. You can please contact our digital team and we'll be able to arrange a demo for you. So then I guess just to wrap up, thank you to everyone, all the attendees for joining, and of course thank you so much to our panelists for taking the time out of their busy schedules to be able to come here and to share their perspectives. As always, it's a pleasure talking to you, and thank you very much. We'll speak soon.
All right, thanks a million.
Thank you.
Bye everybody.